I'm Larry Boyce, and I'm coming to you from Edinburgh. We're at the Myeloma 2017 meeting, and we just finished our session on model systems. And model systems are really important in myeloma research. We know what's going to happen in the patients, or trying to understand what's going to happen in the patients in the laboratory will help us in developing new therapies and understanding how our current therapies work. And we have three of the speakers from the very exciting session here. Dr. Ocio, Dr. Shain, and in the middle, Dr. Chesi, who gave these very interesting talks. And so we had very different types of models that we talked about. So why don't you all tell us a little bit about the model you discussed and maybe some of the highlights of it. So Ken, why don't you go first?
Sure, thanks so much, Larry. Yeah, the model we've kind of developed at Moffitt with our colleagues is looking at an ex vivo model system where we actually take patient specimens and we can try to use them to actually assess how a patient may respond in real life to a different set of drugs, whether it be drugs we use in myeloma today or in therapeutics of the future. And so it's our way of using the patient itself to try to model what we can do in terms of learning as well as predicting outcomes in those patients.
That's great. And the other two talks were really about using animal models or mouse models. And so why don't we hear a little bit about the types of models that you talked about and what we've learned from them. So, Dr.
Yeah,
Chesi?
well, I use a completely different approach in a way because I go entirely with on mouse tumor, and I think as the advantage that we can manipulate easily the genome of the mouse, so we can interrogate in vivo what is the function, the contribution of myeloma pathogenesis to a mutation that we identify in human patients. So we can really isolate that mutation, put it in mouse myeloma, and answer the question: is it accelerate the disease? It changes phenotype? It make it respond differently to drugs?
So using mouse models to really learn how the disease develops,
yes.
But we can also use them for treatment. And I think Enrique talked a little bit about those.
Sure. Yeah. What I have tried to do is to review what is the current situation of using models of myeloma murine models that in which we can use as a carrier for human tissues, for human myelomas. And then I have reviewed what are the advantages and disadvantages of all these models, how we can create a human environment in the models, or how we can create an human immune environment in these models, just to be able to test and to see how the drugs work and if they really work in the different settings we have.
Yeah, I think, Ken, in your talk you really made that point about that we're really trying to forecast how things are going, sort of like how we forecast a hurricane and map how it goes. And so can you talk a little bit about how your model allows you to do that kind of forecasting?
So one of the things that we've done is when you forecast, you need lots of data points or data elements. And so we try to create a model, one, where we can keep patient specimens alive for a very long period of time, long enough to catch lots of data points, meaning can we watch them live or die with given therapy. And by doing that, we can actually capture enough values, we can actually predict how many populations may exist that are sensitive or resistant or variants between those two. And then using mathematical models, they can then extrapolate predictions in terms of how is the patient going to respond to that drug over the course of the next several months. And it's not a simple process, but it's one that really allows us to kind of think about what's going on in that patient and that sample at the time that's contributing to response to a therapy in a patient versus the in silico, which is what we're running. So we've built it in a way that we can run 31 or actually up to 127 different clinical trials in silico in a given patient specimen, which is a pretty profound thing when you think about a patient getting one trial at a time in real life. And then to prove that or to validate that, we can then match the drugs they do receive with the outcomes and demonstrate that there's similarity or identity between the two outcomes. We can validate in that setting. So I think a pretty good job of doing that so far. And I think it's even more exciting when we think about what our patients are linked to. So all of our patients have linked molecular data. So every patient we do this with also has RNA sequencing done, also has whole exome sequencing. So over time, we can actually take our patients and say, to drug X, you have sensitivity and resistance, and we can actually look for the molecular profiles that may actually correlate with those two things in silico, and then hopefully translate that again in patients themselves, or maybe to be proved in more specific mouse models, et cetera, of disease biology.
And so thinking about that, in the mouse models, how can you use your models to not necessarily personalize medicine to an individual patient, but to learn things about new therapeutics that we could test and try in these models? Could you comment on that?
Well, I spent quite some time, in fact, trying to utilize the mouse that have the advantage of being in a whole organism. So the immune system, the microenvironment, everything is one species, so is a whole. And answer the question is, can we predict, in fact, which therapeutic would work in the clinic by testing them in the mouse? And we came up with a positive and a negative predictive value for our mouse model. So for example, we tested in this mouse model a variety of drugs for which we have known clinical activity because of we ran clinical trial in patients, and we tested how these agents perform in the mouse, and so we came up with a pretty good predictive value. So agents that work in our mouse model have now 96%, 75% chances, I was an optimist, that they would work in the clinic. And importantly, agents that don't work in this mouse model have a 92% probability of not working in the clinic. And I think perhaps this negative predictive value is quite important, because I think we are not going to waste time in the clinic putting patients to clinical trial with agent that is not likely to succeed.
So along those lines, some of the negative have also been related to the differences in the biology between the mouse and human. And so maybe, Enrique, you can comment on some of the drugs where we haven't been as successful in using a mouse model of the disease, where the other xenograft models have been useful.
Right. So I think the key point in the models, we know that they are models, so is to make them as human as possible, as similar as human as possible. Of course, we cannot get that 100%, because at the end you have to go to the patients. But of course, we have a quite high predictive value of the models, testing new combinations, testing new agents. Some of them we know that they work in the mice. That is, we would say, quite friendly environment for the drugs, and they will not work later in the humans. But I think this is the first step to see if in this, as Marta said, in this environment with a whole human system, a whole system, in this case murine system, but as human as possible, to see if it works really. And then we can also get insights into the mechanisms of this activity, or the resistance even. So I think these are models, we know, but important for going ahead into the clinical practice.
Mm-hmm. And would you like to speak a little bit about the challenges of using the models? I think, as Enrique said, they are models, and none of them are perfect. And so, are there areas where you think you can see improvement or additions to your models to make them better? And I think maybe, Marta, you can talk about, there's one class of drugs that did not initially work in yours.
Yeah, so a class of very important drug are this is called immunomodulator. Thalidomide, lenalidomide, pomalidomide, and now new generation drugs we heard at this meeting along this class are coming along. And there is an example of the limitation of the murine system, because we now know that the receptor for this drug, that is a protein called cerebron, in the mouse has a slightly different sequence, so it doesn't work. And so we put some effort into generating what we call a humanized mouse, because we changed the cerebron, we removed the murine cerebron, and we put in the human cerebron protein, with the hope that we can now finally test this drug, this class of drug in our model.
That's great. And Ken, you also talked a little bit about the end of some of the types of drugs that you're looking forward to actually testing more in your modeling, provided some data on that. And so would you like to comment on how you're going to change your model to test some of the newer immunomodulatory
Absolutely. So we built the model with to try to take into account as much of the tumor microenvironment as possible, mostly what we call a supportive microenvironment. And so a lot of things about immunomodulatory drugs, these new checkpoint inhibitors or antibody therapies that really depend on not just direct toxicity of the myeloma cell, but also the tumor microenvironment, the immune microenvironment, to help them work or to how they function, is really something we have to start thinking about and adding back in. So right now, I would argue it's a limitation of our assay system, but the system's built in a way that allows us to add back. And that's the next step for us in terms of going forward, is how can we then recapitulate this model with a little bit more of the tumor, like the immune tumor microenvironment, not just the supportive microenvironment.
And Enrique, in the models you talked about, there was also some very sophisticated ways to build those in through the use of scaffolds and bone chips. And could you comment on how that's influencing the responses in these
Yeah,
models?
I think that's important because we have been saying that models are models, mice are models, so we have to try to humanize them. And now more and more we are having, as we have said, immunotherapeutic strategies that are now probably the present and the future, and we have to try to have as much as possible the human environment. This we are using with different chips, I would say, of human bone, or even what is, I think it's more important now, is to have this humanized model with human immune system that allows the testing of these novel immunotherapeutic strategies.
Great. Well, thank you again for a great session, and thank you from Myeloma 2017. Thank you very much.